In this video, we will discuss about a secondary reason that could cause the change in pigmentation among the peoples. That is the relative expression of genes in the melanocytes. So initially, in order to test this hypothesis, uh, the people have collected the foreskin from different people from random peoples, and then they did this uh, Fontana medicine stain. It will stain the melanocytes. So what they have identified is that the lightly pigmented peoples, the melanocyte in the epidermis layer is low compared to the melanocytes that are present in the darkly pigmented skin. So from this, this is in line with our hypothesis that darkly pigmented peoples has more melanin content. And later on, they also did this fluorescence imaging. In the bright field imaging, they can see that the amount of melanocytes are low, and same thing they can see in the immunofluorescence image. In this case, the blue color represents the dye, whereas the PM green and red color represents the gene that are involved in melanocytes. In the darkly pigmented skin, they can see the high expression of melanocytes and the more expression of the genes that are involved in this pigmentation. So from this, uh, they made a they this observation supports their hypothesis that in the darkly pigmented skin there could be a relative high expression of the genes that are involved in melanocytes compared to the lightly pigmented skin. Later on, what they did is they did the bioinformatics. In this case, what they did is they did the principal component analysis of the RNA-seq data to see the to check the check the expression of genes that are involved in melanocytes, and then they compare it with the melanin content present in those patients, and they also then connected these results with their ancestry information. So they have find out that most of the African American peoples has high expression of the RNAs that are involved in melanocytes, and similarly they have high expression of melanin. And after that, the Asian and European peoples has low expression of uh, melanocytes, and they will have low expression of these RNA-seq these RNA that are involved in melanocytes. So this further substantiate their hypothesis. Later on, they compare the melanin contents in different uh, ethnicity. So in the African Americans, the melanin contents are high, and they are even much higher than the self-reported ethnicity. And this difference between self-reported ethnicity and the melanin contents is exist for all the three cases. However, the relative expression of melanins, we can see that is low in the case of Asian and European compared to the African American. So this so far it all the results looks like in align with their hypothesis that there could be relative uh, differential expression of genes in the darkly pigmented people and lightly pigmented people of the melanocytes. Then they did the Spearman correlation coefficient to find out the significantly upregulated and downregulated genes that are involved in melanocytes. And when they check these results with the literature, they can find some of the genes that are involved in some of the genes are reported in the literature that are involved in melanocytes upregulations and downregulations. And later on, they represent some of these genes among all the patients in the panel F. In this case, they can see that the high melanin contents high melanin contents is also related to this increase in the wash CH4, and similar is the case for this COMMD3. KF6 and CHD8. And from this kind of analysis, they made a point that some of those genes that are involved in this dark pigmentation skins, and they are able to find out some of the unidentified genes that are involved in dark pigmented skin, whereas some results are some of the genes are already reported in the literature. So that will not only support their uh, hypothesis but also support their approach, as some of these results are in line with the literature. Then they did the genome-wide associated studies, and they pick out the genes of the lightly pigmented peoples and the darkly pigmented peoples, and check which of the genes are low expressed in lightly pigmented peoples and high expressed in dark pigmented peoples. And they can see that the genes that are expressed in highly genes that are expressed highly in the dark pigmented peoples, and some of those genes are expressed low in light pigmented peoples. And same thing they find out in this bioinformatics also. For example, this golga 8am is expressed highly in dark pigmented peoples and expressed low light pigmented peoples. So this could be one of the genes that are involved in this pigmentation of the skin. So this is just a brief overview how they did the bioinformatics. If you need to understand in depth about how they did this second relative gene expression bioinformatics and whole study, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.